Welcome to Biosumit friends. Today we learn about conjugation. Now in one of my previous videos we have discussed about the genetic recombination in bacteria, about the very basics of genetic recombination in bacteria, right? Now today we will learn about conjugation. So first you can see in 1946 Joshua Liederberg and Edward Tatum showed that the bacteria undergo conjugation. Okay. Now this is a process you can understand of transforming genetic information from one bacteria to another bacteria. Okay, now it's mediated by plasmid. Plasmid is a circular piece of DNA, okay, that can replicate independently from its cell's chromosome, the main chromosome, right? And it involves direct cell to cell contact, right? So the main thing is the donor cell will give the F factor, okay, or F plasmid to the recipient cell, and the recipient cell will be F plus 2. F plus means the strain which is containing the F factor is F plus, and which is not containing the F factor is F minus. And after considering or getting the F factor of the copy of the F factor from F plus, a F minus strain will be 1 F plus 2, right? In simple word, F minus is getting the F factor and becoming F plus. Okay. Now what happens in gram negative bacteria? This plasmid actually carries a code for the synthesis of sex pili. Okay, sex pili, or in singular I think sex pilus. These are the projections from donor cell surface that actually contact the recipient and help bring the two cells into direct contact so that the F factor can be transmitted, right? And for gram positive, what happens? They come closely and produce some sticky surface molecules. Okay, and after that it can form the mating bridge. We will show one diagram how what is mating bridge was like that, and the copy of F factor is transferred to the F minus. Okay, now it can be of three types, this conjugation: F plus F minus, here you can see F plus F minus, Hfr F minus, and Sexduction. So let's see first the F plus F minus. Now here you can see there are two strains, okay, two bacteria you can say. This is one and this is another. So I am showing this diagram, or showing this diagram after creating this mating bridge. So this is the bridge, okay. So earlier these are two separate, okay, and they come to close, they come close, and they are making some mating bridge to transfer the F factor. Now this is the main bacterial chromosome, okay, and this is as F plus, so it is containing F factor, right? And here you can see there is no F factor, so it can be called as F minus. So after creating this mating bridge, what will happen? The one copy of F factor will transfer to the F minus. Here you can see one copy is going to the F minus. So finally what we will get, the F plus cell is F plus as it was, and this F minus cell is turning into F plus as it was containing one copy of the F factor from the previous F plus cell. So this is the normal F plus F minus conjugation, right? Now coming to Hfr. Hfr means high frequency recombination. Suppose here you can see one F plus cells, right? So it has its own chromosome and the F factor. Now due to some mechanism and due to some conditions, this F factor is getting integrated to its own chromosome. Like here you can see the blue one is its own chromosome and the red one is its F factor, right? The F factor is getting inserted in the main chromosome. So then we can call it Hfr. Now suppose you have learned about the F plus F minus. So if this Hfr is going to have the conjugation with F minus, like this will create a total of copy and it will be transferred to the F minus, then that type of conjugation we can call as Hfr F minus conjugation. Okay. Now what happened? You can assume that earlier the F factor was very small, so it can have the copy and it can be transferred fully to the the previous F minus, right? But now you can see this is the main chromosome plus the F factor, so it is very big. So normally what happens, the chromosome breaks before it is completely transferred, okay, to the recipient cell. So mostly in such cases you can see the part of the chromosome is getting transferred to the F minus, not the full, okay. So this is the Hfr F minus. Now the last part, Sexduction. Sexduction actually includes the F prime F minus conjugation. Okay, so what is F prime? Suppose here you can see this is Hfr, right? How it is being made? The main chromosome is integrated with the F plasmid, or rather to say the F plasmid is getting inserted in main chromosome, right? Now somehow due to some many conditions, if this thing again is getting separated, okay, but this time what happens, the previous the normal chromosome, I mean this its own chromosome, it's not getting separated fully. It can have some part of the F factor, and also the F factor can have some of the part of bacterial's own chromosome with it, so it can be separated in two parts, but both of this part, this own chromosome and F factor, will have some of its partner, or not partner, actually the part of the opposite type of factor, you can say, right? The main chromosome is containing some part of the F factor, and the F factor will contain some part of the main chromosome. So it can look like this. Here you can see this should be the main chromosome, right? But it is getting some red part. Red part indicates the F factor. And now here also you can see the red is F factor, but it is containing the some part of the bacterial own chromosome. Okay, so now it's not like the previous two, right? So these are some types of mixing. So in that case we can call it F prime. Now suppose if it was not getting mixed a little part, then we can call it the normal F plus, right? But this is F prime. Okay, now when it will undergo the conjugation with one F minus, we can call it sexduction. Okay, so again what will happen, this will create a copy. Okay, it will undergo replication and create a copy and send it to the F minus, right? So this is called sexduction. Okay, so in short, conjugation is making a mating bridge and transforming its genetic factor to the other, to the recipient. Okay, the donor is sending the F factor to his recipient. It can be of F plus F minus, like the normal Hfr F minus, when the plasmid is getting inserted into the main chromosome and it's getting replicated and getting sent to the F minus, or it can be sexduction, like it is making the F prime and the F prime plasmid is undergoing conjugation. Okay, so I hope you have understood the conjugation in a very simple way. Thank you.